for those who doesn't know Source Molecular, we are a company that provides genetic and analytic solutions for waters. So we have been known for mostly providing microbial source tracking testing service, but we are expanding, and we are the only accredited lab for MST, and we also have digital PCR, pathogen testing by both cultural method, molecular method, and some other products. Okay, keep going. Next. Oh, looks like I can advance size. Great. So why we're here today? So in 2012, EPA published the revised recreational water quality criteria. In it, the regulatory focus has shifted from fixed numeric standards to a risk-based framework. So essentially, previously, what you need to do is, if your intercoccus geomean is less than 35 CFU per 100 ml, then you're okay. But if you're above that, then you're failing. So this is assuming that the intercoccus indicate human fecal contamination. So that's not always true. We'll get back to that a little bit later. But now the regulatory focus has shifted to public health risk. And as long as you're providing equivalent public health protection, your intercoccus could be above 35 CFU per 100 ml, but still in compliance. So this opens up a door for sites where the sources for fecal indicator bacteria, such as intercoccus, are mostly non-human or even non-fecal. So EPA would allow you to establish site-specific criteria based on something called Quantitative Microbial Risk Assessment, QMRA. So just to understand how this shift came about, let's take one step back. So the reason why we care about fecal contamination, this is mostly because the driver is basically public health protection. So the fecal contamination may contain pathogens, which is a risk for public health, and that would limit beneficial use, such as recreational activity. So currently, the way we monitor for that is through measuring fecal indicator bacteria, such as intercoccus and E. coli in the water using culture-based method. So you grow them up on a plate, and if the geomean for intercoccus is above a fixed threshold, let's say 35 CFU, then it's assumed that the water contains human pathogen, is dangerous for public health recreation, for recreation activity, and a sign will be posted. Okay. So the reason this can be done, this type of monitoring is protective of public health risk, it's because the chain of inference. When you see a high concentration of fecal indicator bacteria, it's inferred that you may have waste in your water, and it's inferred that you may have human waste. And from that, it's inferred that you may have human pathogen, and therefore may infect people and cause diseases. And epidemiology studies are used to connect the concentration of FIB with the illness rate in the recreators. And that's how the recreational water quality criteria gets developed. So the epidemiology studies that was conducted in waters predominantly impacted by human fecal contamination. So the water quality criteria is basically established through epidemiology studies that were done at sites that are predominantly impacted by human fecal contamination. So the epi studies establish a relationship between FIB concentration and public health risk. And so as you can see there, I chose the example for a a 35 Enterococcus geomean will correspond to an illness rate of 36 per 1,000 recreator. So that's how the recreational water quality criteria come about. Next slide. However, not all sites are predominantly impacted by human fecal sources, and your site may be mostly the Enterococcus at your site may be coming from mostly animals or plants or just biofilms. So because Enterococcus are not really specific to human fecal material. Next slide. So because of that, the chain of inference that I showed you earlier breaks down. So you cannot infer from fecal bacteria, from FIB to waste to human waste anymore. Next slide. Additionally, sources also differ in risk. So the same FIB concentration from different sources correspond to different level of human health risk. As you can see from this figure, if at your site, if your Enterococcus mostly come from gall, the corresponding human health risk will be significantly lower than the site where the Enterococcus come from sewage. Next. And because of that, the water quality numeric standards, the numeric criteria that's established based on at the site, mostly impacted by human, would not apply to the site where the majority of the contamination is gall. Next. So the concept of QMRA is to basically establish a criteria that provide the equivalent public health protection. The way you do that is, first, you demonstrate your site, the human fecal contribution is really minimum. And then you characterize your water quality, figuring out what are the different sources at your site, and then measure the pathogens from each of these sources. And then you input all these data into a series of models, QMRA models, so that you can determine the alternative site-specific criteria, basically the alternative Enterococcus concentration that corresponds to, let's say, the 36 per 1,000 illness rate. Next. So this seems pretty straightforward, but how do you actually demonstrate your site have minimum human fecal contribution? And in a scientifically sound and statistically defensible manner, as stated in the 2012 criteria. So currently, unfortunately, there's no guidance or standard procedure on how to do this. Next slide. So this presentation intends to share with you a scientific thought process on how to demonstrate human fecal contamination at your site as minimum. This is not a policy talk. So I want you to keep that in mind. So this talk is also adapted from a guidance document that was submitted to Los Angeles Regional Board in 2016. So the document was prepared by me along with Drs. John Griffiths, Steve Weisberg, in consultation with the LA Regional Board, based on the collective scientific understanding of the field at the time. I also incorporate some new scientific advances since 2016. For example, since then, a standardized data interpretation approach called Human Fecal Score has been published. So the data acceptance criteria for MSD data has also been published. And a new human fecal marker crossphage has been published as well. Okay, next slide. So this scientific thought process involves 4 key areas of consideration. The first one is, what is considered minimum human contribution? So what does it mean your site is non-human enough? And the second area of consideration is, how do you actually measure the human fecal contamination? What markers should you use? And what technology should you use? Then third, what about sampling design? Would 10 samples be enough to show that you have little human? How about 10 samples from the same day at the same location? And then finally, what are the requirements for lab analysis and data analysis? How do we use standardized protocols and control the quality of the data? Next slide. So first, what does it mean non-human enough? Here, the first problem we have is there's actually no threshold right now. EPA may have speculated using 10 to 20% human as the threshold of too much human. So risk simulation that was published in 2014 also showed that perhaps if less than 10 to 30% of your culturable Enterococcus comes from human source, perhaps your site can be considered have low enough human contribution. However, there is no official number, no official threshold at the moment. We don't know whether it's 10, 20, or 30%. Ultimately, this is actually a policy decision involving more than just scientific input. So just for the ease of communication in this presentation, let's assume we're going to use 10% as the threshold, okay? If you have more than 10%, that's too much. If you have less than 10%, then that's not human enough. Next slide. But then here comes the second problem. 10% of what? What's the interpretation of this threshold? The ideal interpretation is based on the risk simulation I just mentioned. Ideally, it should be interpreted as less than 10% of the culturable Enterococcus came from human fecal material. And this means that you need to be able to assign the portion of the culturable Enterococcus to the human source and other sources. The reality is we cannot do that type of source apportionment with the technology we have today. This is mainly because the human fecal contamination is measured not by Enterococcus. It's also not measured by cultural method. Okay, next slide. So what are the options we have? So first, perhaps we can interpret from a magnitude perspective. Let's say if you have less than 10% of the amount of fecal contamination that came from human, perhaps we can say your site is non-human enough. So that could be 10% of total mass of fecal DNA, total fecal DNA, or total Enterococcus by qPCR. However, the feasibility and the appropriateness of this approach is uncertain, and research is still ongoing. So this approach may not be attainable at the current stage. Next slide. And then the second option, what about interpreting by frequency? Let's say if you have less than 10% of the samples that are positive for human fecal contamination, then perhaps we can say your site is non-human enough. You may move forward with the QMRA to establish site-specific criteria. So with this approach, you need to define 10% of what number of samples. 10% of the total number of samples is what we would recommend. Another number of sample will be 10% the number of samples exceeding intercoccus criteria. However, that approach is not recommended because, as we know, particularly at a site that you're trying to consider QMRA, you by definition would have mixed sources. So the intercoccus concentration and exceedance would not correlate with human fecal contamination at all. So total number, 10% of total number of samples is what would be recommended. You also need to define what would you actually call a sample positive for human fecal contamination. The bottom line is this definition will need to be based on scientific. It would have to have a scientific basis. Okay. So next slide. And a third option would be, how about we benchmark against a reference site? So as I mentioned, a human fecal score has been developed. It's basically a standardized mathematic way to consistently quantify the extent of human fecal contamination at a site. It gives you one single number as a score. So the higher the score, the more contaminated you are with the human fecal material. So if you can demonstrate your human fecal score at your site is less than the score at a reference site, perhaps your site has low enough human to be eligible for QMRA. Okay, next slide. So in summary, ultimately, this really is a policy decision based on the best available science and also the best data from the site under evaluation. So my recommendation would be that the stakeholders take the initiative to obtain the best study design, the best data, and provide regulators options and multiple lines of evidence to help them enable regulators to make decision. Next slide. So now moving to the second area of consideration. How do you measure human fecal contamination? First, which marker to use? There's several good markers out there based on evaluation studies. The marker of choice would be HF183. This is the best marker that has been evaluated in many evaluation studies. So, and the second question is, how many markers do you use? If you have limited resources, I would say just use HF183, just this one marker, as resources are better spent on improving other study design elements, such as increasing the sample size. However, I do want to mention the motivation behind using multiple markers. They're twofold. One is you may be afraid that the HF183 marker is not picking up human fecal contamination all the time, because it is known that this marker is not present at all human populations. Okay, and then on the other hand, when you may suspect that the very low concentration of the H183 marker could result from cross-reacting with some animal sources at a very low level, you may want to have a second marker to confirm your HF183 results. So if you have the resource, then go ahead and use two markers. Next slide. Then now you decided to use HF183, what method you actually use. So I'm not going to go into the technical details of comparing qPCR and digital PCR. the bottom line is digital PCR is superior in performance. So if it's available, that would be my recommendation. However, qPCR, it's a lot more labs have qPCR equipment. It's more widely available. And also, for both methods, standard protocols have been published in the scientific literature. But on qPCR, its protocol actually will be published in the Federal Registry as a national standard method. So that's another advantage. And the bottom line is, if you find a reliable lab and also get reliable reference material, either method should be fine. Next slide. So now the third area of consideration, sampling design. How many stations do you want at your site? Ultimately, this depends on how widely you want your site-specific alternative criteria to apply to, how wide of area you want this to apply to. US EPA basically says that the site-specific criteria can be applied to a water body that shows uniform water quality. So that means that if your site have very high spatial variability from one location to another, then that's an argument for having more sampling stations. If you have little spatial variability, the water is quite uniform, the quality is pretty well mixed, then fewer stations would do fine. Next slide. Then in terms of how many samples you actually take in one day and when you go out. So once a day is sufficient. That's kind of very common in most recreational water sampling activity, routine sampling. And you should also go out in the early morning to avoid a lot of confounding factors. I'm not going to go into detail there. And this is also a very commonly done in routine monitoring, even with just the FIV monitoring. Next slide. Then now, how many samples do you actually need to demonstrate that your site has a little human? You need to have enough sample size such that you have sufficient statistical power to prove your human contribution is low enough. For example, you take a bunch of samples from a site. If you were using the frequency interpretation, you calculate that 9% of your samples were positive for H1N3. But remember that the 9% is not a point, right? It has an error bar because you estimate it based on your sampling event. So you need to collect enough sample so that the 9% plus minus error bar is statistically significantly less than 10%. Similarly, if you're using the human fecal score approach, the human fecal score has a confidence interval based on your sample size. And you have to collect enough samples so that the human fecal score is statistically significantly lower than the score at the reference site. So as you will immediately realize that this will depend on the temporal variability at your site as well. So if your water quality varies highly from today to tomorrow to the day after tomorrow, and then you will need more samples to fully characterize your site. But if your site water quality is fairly stable from day to day, which means they have low temporal variability, then perhaps you don't need as many samples. However, in most cases, prior knowledge like this, it's not available. You won't know until you actually start sampling. So based on simulated data and also examination of real data from Southern California sites, a 75% coverage is recommended. What that means is that if you're interested a period of 100 days, so that's your period of interest, then you should sample at least 75 days in order to capture what your site is like during the period you're interested in. Then finally, do you choose more samples or more replicates per sample in the lab? So for resource optimization, if you have limited resources, always go for more samples. Next slide. Last but not the least, the lab analysis and data analysis. So it's very important that standardized laboratory protocols is used, and not only on how the qPCR or digital PCR is performed, but also on data QA/QC. This ensure consistent, high quality human marker data. So as I mentioned before, data acceptance criteria has been published, so that should be followed as well. And we also need to pay attention to lab accreditation and automation. So far, what we have covered is how to demonstrate if your site have low enough human contribution so that you may consider QMRA to establish site-specific criteria. So that's what's kind of included in this orange circle here. So it's only one part of the bigger approach that you need to solve the FIB problem. So in reality, what we talked about won't be sufficient. So you actually need to start from the top where you have the FIB problem, then go down the flow chart I'm showing here, and hopefully to reach a resolution to resolve your problem. So what do you actually need along the way? Next slide. So I tend to call this a three-legged stool approach, pun not intended. So basically, starting from the top, if you have a FIB problem, the first thing you usually do is let's do micro-resource tracking or sanitary survey and micro-resource tracking, trying to find out what source, what's causing this problem, what source you have there. If the micro-resource tracking results is showing that you have a lot of human there, then you go down the right side that you remediate the human sources using various BMPs. Then you evaluate the effectiveness and of this remediation. And if after that, you are meeting the FIB criteria, then yes, you reach a resolution. But if you're not meeting that criteria, you need to go to the left side. So another way to reach the left side is if your micro-resource tracking work, like we just following the proper study design and other consideration is demonstrating you have minimum human, then you can go down the left side to pursue the QMRA. So the three-leg stool I'm mentioning is you need to first have a local partner, like a consulting firm or something, or someone to help you organize the entire project, communicating with the regulator. And then you also need to have a reliable lab to be able to perform the micro-resource tracking testing, to also characterize the sources and doing the pathogen testing in the sources, because these data, the pathogen data, is needed for QMRA. Then finally, the third partner you need is a reliable firm or reliable researcher who can do QMRA modeling. And then from the risk modeling, if that shows you are providing the equivalent public health protection, you may be able to establish a site-specific criteria, again, with your partners interacting with the regulators and reach a resolution from there. So next slide. Yeah, with that, I'd like to thank you all for your attention. And I'm very sorry about the connectivity issue earlier and starting this late.
How does HF183 compare to phylochip?
So these two, actually two very different technology. H183 is one single marker that you can quantify. It's quantitative. You can the results you are getting will be the number of H183 gene copies per 100 ml of water. Phylochip is a completely different category of a microresource tracking tool. So it will not give you a quantity. It's semi-quantitative. So if I can send you some more conceptual reading. I've written a paper on community analysis approaches for MST, which include phylochip. Phylochip is one of the community analysis approaches for microresource tracking. Yeah, if you send me an email, I'll send you that.
Hi, this is Akin Babatala, City of Santa Cruz. I don't know if I'm unmuted yet.
Yes, you are unmuted.
I can hear you.
Okay, thank you very much. And thank you for a very eloquent presentation. And you may not know, but we have a major beach here where we have applied molecular markers to both describe the extent of the problem and to characterize using more than HF183, but also HUM2 and HUM3, somewhat consistent with your idea that the cross-reactivity issues with HF183 might make results less definitive, and that was our approach. Now, after having analyzed approximately 900 samples over 2+ years, the data shows that most of the sites, and we picked 9 strategic sites, have less, in fact, have nothing detectable, and 3 sites, one has about 13% frequency of detection for HF183, and other sites have 8 and 7% and thereabouts. What would you say if an independent analyst came and ruled that on the basis of those low levels of detection, and I didn't even mention that the copy numbers are somewhat ridiculously low, usually about 2 to 3 times detection level. But if an expert came around and said, you need to do QMRA, what would you suggest?
An expert come about to say you need to do QMRA?
In spite of those numbers, and consistent with what you've said, which is basically if you use, generally speaking, a frequency of detection of 10% as a good threshold, and presumably assuming that you have low copy numbers, that would not be a good place to spend your resources. What would you say to that? Because I just described a scenario where 1 out of 9 sites had a 13% frequency of detection in 900 analyses over 3 years, almost 3 years. And every other site was either non-detect or sometimes at 8%. And copy numbers are maybe 100, maybe 300, thereabouts. And this is digital qPCR, by the way, not even PCR.
Oh, okay. Yeah, that makes sense. 900 samples from one sampling location, or
from a total of
quite
9 sites within a 300, let me say 600-foot area of the beach.
Okay. Well, so if you have like about 100 samples from one location with a digital PCR where it's showing you have very low frequency of detection, I would communicate with the regulator, interact with them and present the data. Of course, having expert, look over the data, making sure everything, the quality control, everything, the interpretation, it's all up to, the standard. And
Yes.
then the recommendation would be interacting with the regulator along, take along your partners, and then have a discussion, basically provide multiple lines of evidence and all the different options. You can interpret the data, like I mentioned, right? How low is enough in the beginning?
Right.
So, because this is actually brand new, so I feel like we have the responsibility to help, the entire community to help move this forward,
I agree.
Hi, this is Matthew Verbila from San Diego State University. Thanks for the presentation, Yiping. It was very, very informative. I was wondering if you can comment on the persistence of HF183 versus viral pathogens or viral MST markers, and your perceptions on viral markers like PMMOV or crassphage.
Right. So the persistency, I will speak. It highly depends on the site condition. So that's first. Then second of all, so crossphage, I don't know how many, in the field, in situ like degradation study has been done. So I can't compare there. PM, the mild pepper model virus. So it's known to be pretty resistant to a lot of stresses. Again, I haven't compared those two, but just generally speaking, being a bacteroidetes that doesn't like oxygen, and also being a bacteria, it's probably, if I have to guess, it's probably less persistent than the viral MST markers. Does that answer your question?
Yeah, thanks. Yeah, our lab is getting into the use of PMMOV and trying to do some comparisons with HF183 persistence.
Yeah.
But we hadn't thought about crassphage, and that's one thing that was of interest.
Okay.
Can you say anything about the detection levels that feed into the percentage? Example, what is a meaningful level?
That's a good question. So on the slide, I had mentioned that the definition of positive, I think that's what you are referring to. So right now, so among the entire MST research field, or even just practitioners, there may not be a consistency. So some people may choose to say any amplification is of interest. We want to be conservative in terms of protecting public health risk. So that's what we're going to use. But another philosophy would be that, well, if they're that low, that can't even be quantified, maybe we shouldn't count that as positive. But regardless, it should have a very solid scientific basis, whichever decision you choose, and should also, again, interact with your entire team, including the regulators, to get everybody's consensus as well. So, and this also plays into the objectivity in interpreting MST data. So one of the things that happened before is usually the MST data is interpreted through a process called best professional judgment, where you hire available best professional to look at the data, then they give you their interpretation. So we have done some research before. It turns out there's a lot of variability from one professional to another professional. We had asked 10 experts who have done MST research or implementation for a long time to look at the exact same set of data, then ask them to rank exact same set of data, including data from 26 sites. So we asked the expert to rank the 26 sites based on their interpretation of the data in terms of how polluted the site is with human fecal contamination. And then we used correlation on the site ranking to assess how consistent the professionals are. So the correlation was actually from minus 0.33 to 0.97. So the conclusion from that study is the best professional judgment type of interpretation is highly inconsistent. So, and that motivated the development, the team, that motivated the development of the human fecal score that was developed by a team including USEPA researchers from Stanford and from SCORP. So, and the human fecal score is an objective approach that doesn't need to kind of have a subjective definition of positive. It basically takes all data, including not amplified regardless how big the signal is, and then compute it through a Bayesian approach and give you a score that reflects the extent of human fecal contamination. So that's a long answer to your question, but it's a very good question. There is a certain level of subjectivity in some of the interpretations. So that's why as the field moves forward, I think the standardized, consistent interpretation of MSD data is very important.